Hello, everybody. Welcome back. This is another video. This time we'll be discussing a microbiology. This is from Wrocław Medical University. I'm just reviewing over our lecture 2 for medical microbiology this semester. So I just wanted to make sure that you guys get exactly what the professor explained about these things that she presented in lecture 2. First, we're going to talk about pathogenicity and the virulence factors. Pathogenicity is ability of microorganism to cause a disease. Sometimes we can call a microorganism non-pathogenic because it can exist within you but not cause a disease. But if it causes a disease, then it's pathogenic. Virulence factors are factors that help this bacteria or a microorganism to affect you in a way, hence cause diseases. So these virulence factors extend from toxins to enzymes to adhesins that are attached to its membranes, things that are also released, in this case, toxins or enzymes, and they induce different kinds of diseases to your body. The two types that we know of the bacteria that exist, exogenous and endogenous. So exogenous comes from outside, exists outside, and then goes inside to cause you diseases or infections, and these are called exogenous infections. Endogenous bacteria are that ones that lie within, let's say, your intestines or even on your skin. And so if they affect you, they can cause endogenous diseases. And microbiome is an example of an endogenous bacteria. They're either resident and transient, so they will be inside, resident as in inside your intestine or inside your body, and transient is on the skin. And so if they infect you, they get into the blood or some kind. If they infect you and get into the blood around the circulatory system, they can cause diseases that are called or categorized as endogenous diseases or infections. Systematic dissemination disease. It just means that disease can go to different organs. And so dissemination is just a spread of the disease, and it happens by the blood. Here, in this case, we've discussed already, just a summary of what we've discussed so far. We're going to start very quickly, however, very specifically, and I hope you guys can start taking notes from this point onwards, because things can be very confusing. But if you study this well and sound, I think you'll be fine. So we started with this family called Micrococci. It has two genuses within it, Micrococcus and Staphylococcus. The Micrococcus species that they want us to focus on is Micrococcus luteus, and the Staphylococcus contains three main species that we're going to study: Staphylococcus aureus, Staphylococcus epidermis, and Staphylococcus saprophyticus. Okay, now we're going to go on to the second family that we're going to focus on in this lecture, is the Streptococci. I think that's how they pronounce it. This word, it's pretty hard. My tongue gets stuck sometimes. So the two genus that are present within this family is the Streptococcus and the Enterococcus. Streptococcus is made up of four species: Streptococcus Pyogenes, Agalactia, Pneumonia, Orale. And with the Enterococcus, you have the Fascialis and the Facium. And so these are the main ones within the genuses. All right, so Staphylococcuses, they are, this is just one way to differentiate or find different bacteria within the Staphylococcus family. This is just one small field of what we're discussing from the whole image. Coagulase basically is an enzyme that causes clotting. And so those bacteria, if they release coagulase, they'll be able to form clusters that they can grow in, and the body cannot reach those bacteria because they're surrounded by a protective clotting membrane. So coagulase is an enzyme that converts fibrinogen to fibrin, and this fibrin is a sticky material. And so a lot of those bacteria that secrete this enzyme are able to collect themselves in a clot within the body and grow and divide and secrete harmful substances. So coagulase enzyme, a bacteria can be either positive or negative. Negative as in it cannot produce this enzyme. So coagulase positive means that it can produce. So the one that is able to produce this is Staphylococcus aureus, and the one is not is the Staphylococcus epidermis and Staphylococcus saprophyticus. Let's start with Staphylococcus aureus. In this slide, the professor just categorized those things that lie on the bacterium or is released by the bacterium. So the ones that lie on, and these things have to all be memorized for every bacteria, whether it's released or stuck on, so that you know exactly how this bacteria functions. So Staphylococcus aureus, things that are attached to its membrane and help it to cause its effect. First, the A protein, and A protein is the one that prevents this bacterium to be caught or identified by the white blood cells, hence prevents phagocytosis. Coagulase, I've discussed the function of this being mainly producing clots of aureus to help them grow and prevent the body from destroying them. Peptidoglycan, this is just a structure that is within the cell wall of the aureus, and in this case, it's gram-positive bacteria, so this layer is very thick, and so it just helps to protect it. Capsule and adhesins, these are things that also help it to stay invisible. Adhesins are the ones that help it to attach to things, and so prevent body fluids from moving it from to different regions. So it wants to stick to one place to cause infections. Staphylococcus aureus would also release things such as coagulase. So yes, in case of coagulase, it can be stuck to the membrane of aureus or released to cause effect elsewhere. Catalase, here this enzyme breaks down hydrogen peroxide, and so hydrogen peroxide is a very harmful one for aureus. So it uses catalase to break down this hydrogen peroxide, and so you can see that it's able to bubble off some oxygen as it breaks down hydrogen peroxide. Enzymes that are released by aureus is hyaluronidase and fibrinolysin. Hyaluronidase breaks down hyaluronic acid, which is just a substance within our body that helps to stick our body cells together. So this is very useful as it breaks this hyaluronic acid structure and gets in between cells so that it can hide from white blood cells that can break it. And fibrinolysin is an enzyme that brings down fibrin, I think. And so this enzyme is very useful for aureus to infect a wound. So you know how wounds need fibrin to blood clot and prevent loss of blood. If aureus is present, it would infect this wound and cause it to not stop bleeding. Toxins here, this is the poisonous toxins that were released by aureus. TSST, it's the toxic shock syndrome toxin, which causes a 10-time effect of immunity, and it can cause a lot of inflammation and basically a shock to the body, as I think it'll cause damage to tissues. Hemolysin is able to break down what is hemolysin? I really forgot. I'll just hemolysin, just give me a second. But let's discuss leukocidin, Panton Valentine here. Leukocidin, from the word leukocidin, we can understand that leukocidin is able to break down white blood cells. And exfoliating toxins, this is a toxin that helps break down or separate the epidermis from the dermis. And so you can see that Aureus can cause infections to the skin that are very dangerous. Enterotoxins here, enterotoxins are used to break down or damage cells within the intestines, such as enterocytes, and so absorption function of the intestine is destroyed. Hence, nutrients are not able to be broken down, and so you can find that the diarrhea is formed or loss of a lot of water due to damage of both enterocytes within the small intestine and the large intestine. Hemolysins, yes, right now I remember, is a enzyme that breaks down lipids and proteins that causes lysis of red blood cells, and so disruption of their membranes, basically. So it can cause so many effects within the body, and we're going to discuss diseases in a bit of Aureus, but mainly what Aureus does is it would damage the skin, and so the more superficial, it will cause more superficial things, so like carbuncles or furuncles. But if it goes deeper, it can cause things like cellulitis. And if it reaches the blood, it can cause many other things that can be very harmful to us. And I'll show you guys in a bit. And so in this structure, in this presentation, we can see that Aureus is able to protect itself by protein A, which prevents immune activity cells towards it. Here, this is how coagulase works. The bacteria would release coagulase, gets the other bacteria to clot around each other. This produces an environment in the body for itself to replicate, and then it would definitely release something called kinase that breaks down this clot, and so it will set those new bacteria free to allow it to infect other places. Super antigens, it's just here for this slide, we're just defining what a super antigen is and what are some very famous super antigens present and released by most of the bacteria that we're going to discuss in this presentation. All of the bacteria that you have seen do not release all of those at once. They may release one or two because they're not able to do so because they have genes for specific super antigens, so they can't have the genes for every super antigen. Just famous ones: toxic shock syndrome toxin, staphylococcal enterotoxin, staphylococcal exfoliatin toxin, and pyrogenic exotoxin. Diseases caused by Staphylococcus aureus is SSSS. I think this is, let me get the full form of SSSS. It's just a skin disease. Caused by aureus. And it's called staphylococcal scalded skin syndrome, which is a serious skin infection. It's basically the skin would start peeling. Impetigo is another very deep skin disease. Folliculitis, fornicles, and carbuncles. You see all of those things are around skin. And so if the bacteria reaches deeper to that dermis, it would reach blood vessels eventually, and so it would cause things like bacteremia, sepsis. This is where the body's trying to fight bacteria but damage itself. Catheter-related stream infections. It will also cause a shock, and if it reaches the heart, it will cause endocarditis, pneumonia, UTIs, osteomyelitis, and food poisoning. Okay, these are just main diseases caused by this bacterium. Yes, just here saying purulent are pus producing, pyrogenic is inducing fever. Infections, this is what we just discussed. The SSS and TSS, toxic shock syndrome, and the other shock syndrome of the skin and food poisoning are toxin mediated. Purulent means that it's on the skin. And these are endocarditis, pneumonia, and bacterium. We're just identifying disease caused by Aureus into different categories: the purulent ones and the toxic mediated ones. This is the epidermis. This is the action of exfoliating toxin released by Aureus, and that's what it does. That's why it's very harmful for the skin. It can separate epidermis from the dermis. The next two from Staphylococcus, and they're not as big as Aureus, but they're just still harmful. Epidermis, this is the one that is very related to catheter-related diseases. If a catheter is used in the bladder or in the blood, it can be affected by epidermis. And so things such as UTI infections, endocarditis, and stuff like that can happen if the bacteria reaches the blood. Okay, but mainly it's catheters and prosthetic heart valves that where those bacteria love to grow on and cause those diseases. Saprophyticus is a urease producer. It is a urease positive. The difference between those two is that the Saprophyticus is not a catheter related bacteria, but what it does is that it would break down urea, and so breaking down of urea down this pathway at the end would result into production of stones, and they are able to give a UTI infection or endocarditis, and also able to give stones for the patient within their urinary bladder. This is more common among people who are new to their sexual activity, and especially in women. Okay. Next, this is the second family we're talking about. It's Streptococci, very famous for differentiating between its family members using hemolytic activity. There are three groups here that we are going to categorize the bacteria into. First is the beta hemolytic, the alpha hemolytic, and the non-hemolytic. The beta hemolytic, we have two, which is the Streptococcus Pyogenes and the Streptococcus agalactia. The Streptococcus Pyogenes is categorized as group A Streptococcus, and the agalactia is characterized as group B Streptococcus. This is just a Lancefield classification of those two, where they have a specific carbohydrate on their membrane that puts them into these groups. Alpha hemolytic, and just to tell you, beta hemolytic, it means that they're able to fully digest hemoglobin or RBCs to release the iron out of them and use this iron in their pathways, metabolic pathways. Alpha hemolytic, this partially digests the RBCs, or what is called hemoglobin. And so there are two here, orale and pneumonia. And the non-hemolytic, which cannot digest RBCs or hemoglobin, is Enterococci. Hemolytic activity would produce yellow, and this would produce green, and this also appears a little green as well, but yellow here because beta hemolytic activity breaks down the RBCs really well. First one we're going to talk about is Streptococcus Pyogenes. Here, very important that they contain M proteins and hyaluronic acid capsules. M protein is a protein that can be presented on the bacterium, and it prevent them from being phagocytosed, or it can also be released from their membrane to cause autoimmune attacking to the organs of the body. Hyaluronic acid capsules also are found to cover those pyogenes because that would help them in invisibility. Why they use hyaluronic acid is because it is trying to tell the body that I'm part of you, I'm part of the cells that make up the body, because also cells use hyaluronic acid to stick together. So this is just a body substance that is friendly. So it's so clever to use a body substance to cover itself. Toxins released here are pyrogenic streptococcal exotoxin, streptolysin, enzymes, hyaluronidase, and streptokinase. Streptolysin is what causes the, I think the, I forgot. Just give me a second. Streptolysin, I think it's related to the hemolytic activity as far as I remember. Yes. So streptolysin is what causes the hemolytic activity possible here. Streptococcus pyogene is a beta hemolytic, so that's why it's able to break down all of this. Hyaluronidase is an enzyme that breaks down hyaluronic acid that allows pyogenes to hide between the cells and cover itself from the body cells. All right, moving on. This is a way in which M protein can help those bacteria to infect mucous membranes. M proteins can be released and they can bind to fibrinogens. I'm so tired. Okay, and causes degranulation and epithelial damage. This is a way in which M protein can help damage can have those bacteria damage the mucous membranes. Also, this is how we summarize their infections. If you don't know what sequelae is, this is just basically telling you things that happened after the infection. This is not due to toxin or anything. I think it's just due to the presence of antibodies secreted by, well, mainly by those bacteria. And so mainly the M protein. So this M protein would go, or M protein would go into the heart and attack the cells of the heart and bind to the cells of the heart. Then our own immune cells would attack it, thinking that it's a foreign structure. And so this would result in acute traumatic. Glomerulonephritis is basically damage to nephrons because the M proteins are large and they stick into the nephron structures. And so damage can happen here as well. If within a body, the anti-M protein is present, we have a silent infection. If within the body, the anti-M protein is absent, we have local infections, we have generalized invasive infections, and those are very dangerous. This only happens if we don't have antibodies that act against the M protein. If we do have antibodies that act against the M protein, then we have a silent infection. This is what I was talking about, the autoimmune disease towards the heart, rheumatic fever. And this is what I was talking about related to glomerulonephritis. Immune complexes are the main things that cause the damage of the kidney. Streptococcus pyogenes. This is the disease that is caused by pyogenes. Pneumonia is one of the bacteremia, sepsis, septic shock. Streptococcal toxic shock syndrome, and erysipelas. This is just mainly Agalactia. It would, if you hear the name Agalactia, this just reminds you directly of the amniotic fluid, and basically babies are affected by it. Let me just make sure if I'm saying that right. Yes. So if the baby comes out and it's in contact with this bacteria, it can cause meningitis and sepsis in babies. Okay. So that is why pregnant women are tested for agalactia at the weeks of 32, 34. And um, um, if agalactia is present, penicillin is used to treat it. Streptococcus pneumonia, from its name, pneumonia causes damage to the lungs. They exist in pairs, and they're the only ones who exist in pairs. It has a lot of violence factors such as capsule, which is antiphagocytic, and hyaluronidase that digests hyaluronic acid, IgA protease enzyme that breaks down IgA um, in the mucous membrane, causes damage to the mucous membrane. Some infections here, such as the otitis media sinusitis, basically damage, inflammation in the sinuses, pneumonia in the lungs, damage to the lungs. Invasive here, meningitis, sepsis, and endocarditis. Some vaccines that are used to treat against pneumonial effect, such as Pneumovax, and the conjugate there. And this is what I was discussing about agalactia, neonatal meningitis, and sepsis. Blood infections, UTI is also possible. Orale, in the oral cavity, and these are just strains of the oral Streptococcus orale, mitis mutans, whatever it is, and they cause either dental carriers, and if they get into the blood, they cause endocarditis. Okay, this is why hygiene of the teeth is very important. Otherwise, orale will be playing games on you. Enterococcus virulence factors. Yes, so Enterococcus basically is catheter-related blood infections. Also, UTI is a big thing. So it has also many virulence factors here, such as protein binding ECM, lipoteichoic acid for inflammation, hyaluronidase, and some other infections that can be caused by Enterococcus here. This is just a summary of everything that you guys need to probably have in mind, plus any other important thing that you can read from the textbook. And this is also the other ones that we discussed as well. So this is the end of this lecture. I hope you guys enjoyed it. I hope it was useful and remind you of what we did in the lecture. I'll see you guys in the next one.